sexually and asexually. Just for general things, the common things, based on their motility. Of course, taxonomically now, it involves a lot of things, the taxonomically, of course, it includes a lot of the groups and other things. Now, for you as a characteristic group of organisms in the soil, protozoa, of course, you can, they are classified based on their motility organs. Motility organs involved in their motility. Organs involved in their motility. So, into flagellates, then ciliates, then pseudopodiums, amoebae, amoeboids, and lastly, which do not have any one of these motile organs, they are referred to as they are sporozoans. And of course, one more is also there, which are actually found in association with lower things, sectorians, the endoparasites, sometime endosymbionts, and so on. So finally, the protozoa as a characteristic group, they are they are unicellular animals which reproduce both sexually and asexually, and which have holozoic nutrition, means predatory way of nutrition, similar to other animals. Holozoic, they swallow other organisms and digest and survive. And only a small fraction of this protozoa, they have they have pinocytosis. So they they have what are known as food vacuoles. Smaller organic particles are taken, imported into these vacuoles, where the digestive enzymes are produced, where they are digested, and that things, nutrients in turn they are utilized. That's only a small fraction of them have this. Otherwise, majority of them have holozoic nutrition, predatory way of nutrition. Usually, in predatory way of nutrition, predator, so the one which swallows, which kills and eats the other organism, it is called as predator. The one which gets predated, it is called as prey. So predator and prey. Similar to the parasites, parasite, the one which lives on the other organism at the cost of the other organism, it is parasite. The one which accommodates the parasite, it is host. So parasite and host. In this case, it is predator and prey. Prey you call. It is similar to the tigers or lions predating on deers and other herbivorous animals. It is similar to that. And the flagellates, they are the smallest among the protozoa. Protozoa. They use only few whip-like, long whip-like structures called flagella. But the only difference between the bacterial flagella and the protozoan flagella is that this protozoan flagella, it is characteristic eukaryotic flagella, which is 9+1 structure. They carry the protein, they are made up of tubulin proteins, 9+1 structure they exhibit. And these flagellates, they are more numerous in terrestrial ecosystem. So among the protozoa, the flagellate protozoans, the flagellates are more numerous, in soil, terrestrial ecosystem, and they are followed by the amoebae. They are followed by the thing, amoebae, which are they are quite large organisms, move by means of the temporary extensions of the cell membrane. You call them pseudopodiums, pseudopods. That's why they are called as pseudopodians. False organs, false motility organs they are made. Because here, because the cells of protozoa, there is no, they lack cell walls. They don't, no rigidity. They are very flexible, and the cell membrane, plasma membrane, can be extended in any direction. Like so by protoplasmic streaming. That's the thing. Like facilitated by that, and the things in extended organs are called as pseudopods, and the one which move with the help of that, they are referred to as pseudopodiums or amoebae. So, and because the flexible structure, amoeboids, you mean they think, pleomorphic structures, the things, like you find the things, in all directions. Then the next thing is ciliates. So the, different groups of microorganisms present inside the bacteria. So then the fungi, then algae, then protozoa. Okay. So next chapter, of course, we have already completed the microbial nutrition and culture media. Of course, in both theory and practicals, we have discussed this. So how these microorganisms are classified based on their the nutrient requirements that we have already completed. So based on their nutritional requirements, so how various culture media that are used in a microbiology lab are devised. Like you have the media, for example, like the differential media, the selective media, and the enrichment media. And these media which are made in the lab, which may be either selective or differential or enrichment media, they may be synthetic or they may be complex based on the kind of ingredients that are part of these things. Okay, this is about the microbial nutrition, which we have already covered. The next chapter, it is the overview, of microbial metabolism. Overview of microbial metabolism. So in one of the, in the theory class, we have discussed what is metabolism. You are aware, you have learned in your chemistry lab, chemistry, like the uh, chemical reaction, about the chemical reaction where two substrates are combined to yield like an end product, and the one which mediates this reaction, it is called as catalyst, a chemical catalyst. And a similar kind of the chemical reaction which happens in a biological system, it is called as biochemical reactions, where the biomolecules, two biomolecules or more than two biomolecules, so they are they react to yield an end product or a set of end products, and which is mediated by a biological molecule, things which is biocatalyst, which are referred to as enzymes, so the the chemical reactions which happen in a biological system are referred to as biogeochemical reaction. Biochemical reactions. So in living organisms or in a living cell, for example, in case of microbial things, bacterial cell and other things. So in a living cell, you don't come across only one or two, chemical reactions. The thing, you come across numerous hundreds and thousands of different kinds of reactions, which are associated with the oxidative process or reductive process, or which are associated with energy yielding process, or which are associated with the production of various cellular ingredients. You call it that process as anabolism, either catabolism or anabolism. So that particular things, in a cell. So the enzymes, that means to say hundreds or thousands of different kinds of enzymes, you will come across in a biological system, chemical reactions. And a set of these chemical reactions, it is referred to as metabolism. So what is metabolism by definition? So it is some total of biochemical reactions that happen in a living cell. They are referred to as metabolism, metabolic reactions. And of course metabolic reactions are two kinds. They you know the oxidative or degradative reaction, which aims to release energy from a molecule, that is the thing, energy yielding reaction, catabolism. Or the one, which aims to, by utilizing simpler the thing, building blocks, to produce various biomolecules like proteins, carbohydrates, lipids, and so on. So that is the thing, like synthetic reaction, anabolism you call. And all of them together, total of all these reactions, biochemical reactions, mm, that take place in a living cell, it is called as metabolism. And in metabolism you come across two types. The synthetic reactions are referred to as anabolism, which is this anabolic reactions, they are energy consuming. They need energy for this process, they require energy. Whereas the other one, opposite to this, there is another type of reaction called as catabolism, which is actually a breakdown reaction. This breakdown, what sort of breakdown here happens? Oxidative breakdown, oxidative degradation of the molecule happens. For example, simple glucose you take, glucose at the end of the thing, at the end it becomes carbon dioxide and water. It is completely oxidized to carbon dioxide and water. In the process, energy in the form of ATP molecules, it is released. And such kind of reactions are referred to as catabolism. So based on this kind thing, energy yielding metabolism, of course, so broadly there are two types of organisms, the phototrophs and the chemotrophs. The energy, which comes, may come from the sunlight, through the process called photophosphorylation, or from the oxidation of chemical compounds, reduced chemical compounds. So you go by oxidative phosphorylation. And accordingly, the organisms, they may be broadly classified into chemoorganotrophs. Means they are also, the chemoorganotrophs can also be called as organotrophs, or simply organotrophs or heterotrophs. Then chemoautotrophs, it is same thing, with the examples of nitrifying bacteria, sulfur oxidizing bacteria, iron oxidizing bacteria, and so on, all these things. Then photoautotrophs, the one which derive energy from sunlight and which have the ability to fix carbon from the carbon dioxide from the atmospheric carbon. They are photoautotrophs, and the examples are cyanobacteria and related organisms. Then you have photoheterotrophs, and these organisms, they meet their carbon and energy requirements from the organic compounds and light. So that, they are photoheterotrophs are those organisms which meet their carbon and energy requirements from organic molecules and light respectively. So they derive energy from sunlight, but they meet their carbon. Then, the carbon requirements of these organisms is met from organic molecules. So finally you have photoheterotrophs, then photoheterotrophs are photoorganotrophs, one and the same. Photoorganotrophs, then chemoorganotrophs, then or chemoheterotrophs, photoheterotrophs. Chemoorganotrophs are photoorganotrophs, one and the same. Synonymously they are used. So photoheterotrophs are photoorganotrophs, one and the same. Then chemoorganotroph or chemoheterotrophs, it is again one and the same. Then you have again in autotrophs, you have photoautotroph and chemoautotroph. Okay, this is the thing. And all this the examples of the different metabolic groups of organisms that you find, which are found in soil. These organisms, like they derive energy, either from inorganic compounds or organic compounds. Inorganic compounds or organic compounds. Accordingly, so you classify these organisms, auto or heterotrophic organisms. And in these organisms, of course, in any of these reactions, I have explained this in the theory class. So the metabolic reaction, metabolism, anything you call, it's an oxidoreductive reaction. So, oxidoreductive reactions where simultaneously, when one molecule gets oxidized, the other molecule has to get reduced, or when one gets reduced, the other molecule gets oxidized. That's the thing. They are interdependent. When one gets oxidized, the other has to get reduced, or when one get reduced, other gets oxidized. That's the thing. It is an oxidoreductive reaction, not the thing, oxidoreductive reaction. So a reaction where the electrons moves from reduced molecules towards positive potency. And in this process, some organic molecules, they act as electron carriers, are, you also use the word electron vectors or carriers. And some of the examples of these electron carriers are NAD, nicotinamide adenine dinucleotide, or NADP, nicotinamide adenine dinucleotide phosphate, or FAD plus, flavin adenine dinucleotide, or FMN, that is flavin mononucleotide, then coenzyme Q, then cytochromes, various types, cytochrome A, B, C, and so on. Then you have like the cytochromes. Then certain non-heme proteins are also, they also act as vectors. So finally, NAD plus or NADP plus or FAD plus, or FMN molecule, or coenzyme Q, or cytochrome molecules of different types, or non-heme ions. All of them, some of the examples of these, the molecules which act as vectors. So vectors. So what's the main feature of these molecules which can act as vectors, carriers of the electrons, is these molecules can exist in both the oxidized state and reduced state. So you should be able to explain. So the thing, with respect to electrons, because in case of this catabolism, the electrons move from most reduced towards oxidized ones. Ultimately, it is accepted by oxygen. So, for example, glucose most reduced, and at the end, carbon dioxide is formed at the end. So, when oxygen accepts the electrons, it is most oxidized form of things. Okay. So, in this particular case, the electron vectors, for example, NAD plus you take, NAD D plus H, NADH or NADPH, FADH plus H, that is NADP H2, or even these are all the examples. This NAD can exist NADH, or NADH plus H, or NAD. That is NAD it is oxidized state, NADH2 is reduced state, whereas FAD is oxidized state, FADH2 is reduced state. Similarly, NADPH2 is reduced state, NADP is oxidized state, like that. So by having this property, either they can be dual states, oxidized states or reduced state. These molecules will be able to act as carriers of the electrons in the metabolic reactions. And in this process, the things where where oxidoreductive reactions, one acts as donor, electron donor. For example, a simpler this thing, glucose, it is the electron donor. Glucose electron donor, and the oxygen, it is the electron acceptor. Oxygen is the electron. So one molecule acts as electron donor, the other molecule acts as electron acceptor. To be act, to act as electron donor, one has to be in reduced state. Means it should be there. It should have excess electrons. To act as electron acceptor, one should be in a oxidized state. Oxidized state. Based on this particular thing, So ultimately, in case of electron transportation, in the catabolic reactions, the electrons, they originate from the electron donor like glucose, and in the sequentially they are subject to oxidation, and ultimately they get completely oxidized when the electrons having by them, it is accepted by the oxygen. And in this particular case, the electron acceptor which is involved in this reaction, the it may be either endogenous or exogenous. You must remember, based on whether the electron acceptors involved in this catabolic reactions, whether they are endogenous or exogenous in their origin. Endogenous, you mean internally generated. Exogenous, you mean they come from external source. So whether they are endogenous or exogenous, they are broadly classified into fermentation and respiration. So in the first case, if the electrons originate endogenously or internally, that process you call fermentation. And if the electron acceptor comes from outside source, external or exogenous, then you use the word respiration. Further, this respiration, so where the electron acceptor is exogenous or it comes from the external source, it can be either oxygen, where you use the word aerobic respiration, whereas anaerobic respiration, where it is not oxygen, instead of oxygen it is some molecule containing oxygen like nitrate, sulfate, carbon dioxide, and so on. So that's the thing. So based on that, you classify into fermentation, and they broadly, whether their electron acceptor is endogenous or exogenous, you classify into fermentation and respiration. In fermentation, the electron acceptor is endogenous. In respiration, electron acceptor is exogenous. In turn, this respiration is classified into aerobic respiration, where oxygen acts as terminal electron acceptor, or anaerobic respiration, where terminal electron acceptor, it is not oxygen. Instead, a molecule containing oxygen like NO3 or SO4 or CO2, and so on. So that's the things about this. In brief, the information on that, and um, and these electrons which are involved, which are there in this metabolic pathway, they move from most reduced molecules towards the oxidized state, and these electrons ultimately They are passed through electron transport chain. They are passed through electron transport chain, and in the process, a potency is created. A potency is created. So that is the potency from most reduced towards most oxidized. It is called as protonomotive force. This protonomotive force, protonomotive force, it is, protonomotive force. It is created across the double layered cell membrane in prokaryotic organisms like bacteria and archaea, prokaryotes in bacteria. So across the cell membrane, double layered cell membrane, so that happens. Whereas in eukaryotes, so that is thing, that happens. The potency, creation of potency, it happens, protonomotive force happens in the uh, mitochondria, the membranes of the mitochondria, the things, that can be seen, protonomotive force. This protonomotive force is it is responsible for the generation of the ATP molecules. In the generation of ATP, what happens is, in case of this ATP, this adenosine, thing, whether it is triphosphate or diphosphate or monophosphate, three molecules, monophosphate with single phosphate bond, AMP, adenosine monophosphate, or with the two phosphate bonds, it becomes ADP. When with the three, phosphate bonds, it is called as ATP. So AMP, ADP, and ATP. Usually, any reactions, rarely they may reach to the stage of AMP. Otherwise, they usually, like keeps changing between ADP to ATP and ATP back to ADP. That particular conversion you will be able to say. And this particular process, the thing, the addition of phosphate bonds to ADP to transform it into ATP, it is called as phosphorylation. So suppose if the thing comes for a definition, what is phosphorylation? Phosphorylation is a biochemical reaction where an extra phosphate bond is added to ADP to transform, to make it, in order to transform that molecule to ATP. ATP. That's the things, that you'll be able to decide. And in case of ATP, in case of sorry, in aerobic respiration. Hello. In aerobic respiration, you'll be, like you have a separate electron transport chain, etc, which is arranged from most reduced towards most oxidized, that across the cell membrane in cell, in prokaryotes, in bacteria, or the uh, the outer walls of the mitochondria, walls of the mitochondria in eukaryotic organisms. Okay, so in here what happens in the process, metabolic process, usually in catabolism, in aerobic respiration mainly, you have the primary, in the first stage, the larger molecules such as polysaccharides, proteins, and lipids are broken down enzymatically to yield their, to yield monosaccharides, amino acids, and fatty acids. Means they are simpler building blocks of these respective polymers. For example, polysaccharides, they are made up of monosaccharides. The thing, they are broken down to yield monosaccharides. These monosaccharides may be, 5 carbon sugars, or 6 carbon sugars and of different other types like 7 carbons, 4 carbons and co. But more commonly, the 6 carbon and 5 carbon molecules are seen, and that's the thing. 5 carbon or 4 carbon. So the polysaccharide gets converted to monosaccharides. Proteins gets converted to amino acids, and lipids gets converted to fatty acids. In turn, they are further broken down to yield, to yield either pyruvic acid in case of carbohydrates. For example, in the glycolytic pathway, in the first reaction of the catabolic reaction, the glucose gets converted to pyruvic acid. Or in case of proteins and lipids, their broken down products, amino acids or fatty acids, gets converted to acetyl CoA, and they join the reaction acetyl CoA, and further in all the three cases they enter the next stage in aerobic respiration alone. They next they enter the next stage, that is citric acid TCA cycle. Tca cycle, where many intermediates are produced, NADH plus H or FADH plus H, or NADPH2O, or and so on. They are produced mainly in TCA cycle. NADH2 or FADH2 are formed. These intermediary molecules are then passed through the electron transport chain, and and ultimately the thing, oxygen serves as the electron acceptor, and in the process it results in the generation of energy. And a point you must remember here, in TCA cycle, tricarboxylic acid cycle or citric acid cycle, so which are produced, NADH2 and FADH2 are produced, both are produced. But another thing is, you are aware that when NADH2 is passed through electron transport chain, it results in the formation of uh, it results in the formation of 3 ATP molecules, whereas FATH2, when it is passed through electron transport chain, it yields only 2 ATP molecules. What is the reason? The reason is that NADH2, it has very negative potency. It has larger negative potency, and it joins this electron transport chain in the beginning itself, because in the beginning, where the potency is less, in the beginning it joins the electron transport chain in the beginning, far ahead of the FADH2, which joins electron transport chain bit later. As a result, at the end of the thing, at the end, from NADH2, uh, sorry, so the NADH2 yields 3 ATP molecules, FADH2 yields 2 ATP molecules. So this you must remember. For example, if it is given for reasoning, why NADH2 yields 3 ATPs, FADH2 yields only 2 ATPs, the reason is that the NADH2 has more reduced potency, more reduced thing, whereas or it enters the respiratory chain bit earlier at the negative potency, and FADH2 joins bit later at a comparatively slightly positive thing, potency. As a result, they respectively yield 3 ATP and 2 ATP from NADH2 and FADH2. These two things. So the And in case of the catabolic reactions, the complex molecules of, as you are aware, they are broken down, and intermediates are fed to Tca cycle, and finally, and this leads to the generation of precursor metabolites, for the biosynthesis, biosynthetic reaction, and the generation of energy, biosynthetic reactions and generation of energy. The pathways that serve both as catabolism and anabolism are also known as amphibolic reaction. Amphibolic. This is a very important point. So in the catabolic reactions, you have, for example, you have glycolytic pathway, then you have a Tca cycle, and at the end you have ETC, that is electron transport chain. So three components, important components of aerobic respiration. Then you also have other alternate pathways to glycolytic pathway, like you have pentose phosphate cycle, hexose monophosphate cycle. Then you have the internal Dewdorf pathway and so on. All these things are there, and but among all of them, the catabolic reaction, the TCA cycle, which is found in aerobic organisms, it occupies the central stage of metabolism, both catabolism and anabolism, central stage of the metabolism. So one side, for example, when the pyruvate, which is formed from the glucose molecule through the thing, what was that in the molecule? Wait 1 minute. So acetyl coenzyme A, actually, acetyl coenzyme A. Yeah. So they enter, the pyruvic acid, which is formed from glucose, from the polysaccharide side. So pyruvate, which is formed at the end of the glycolytic pathway, it enters through, acetyl CoA to the Tca cycle, whereas they whereas in case of proteins and the lipids, they are broken down products. First initially to amino acids or fatty acids. Further, they are broken down. For example, in case of amino acids, either they undergo both, either deamination or decarboxylation, or in case of fatty acids, beta oxidation. You call. So every stage of the things, step, one cycle, they lose one carbon dioxide. So ultimately, they join the reaction at acetyl CoA, they enter the TCA cycle. In this TCA cycle, starting from the immediate product of the TCA cycle, whether it comes from polysaccharides or amino acids or lipids. So initial thing is, it is the thing, it is citric acid that is formed, and from citric acid, once again, in one cycle it comes back through oxaloacetic acid, through via acetyl coenzyme. Again it comes back to citric acid. In the process, you come across so many intermediate substances, various organic acids are formed, which are actually they act as precursors for the biosynthetic reactions. They act as precursors for the biosynthetic reactions on one side. At the same time, the NADH2, FADH2, which are formed in TCA cycle, they act as thing, they are utilized for the synthesis of ATP molecules. That means to say this TCA, tricarboxylic acid cycle or citric acid cycle, it is, it serves both functions. One side, it is responsible for the generation of ATP molecules. The other side, through the production of various intermediary molecules which are diverted for the biosynthetic pathways. For example, once again, that is in gluconeogenesis. Sometime thing, it is the reverse glycolytic pathway to yield glucose, or the other side, it may be it may go back to the synthesis of amino acids, or it may go back to the synthesis of fatty acids, or it may go back to the synthesis of purines and pyrimidines for the synthesis of nucleic acids. All that happens. So that means to say, it occupies the center stage for both catabolic and anabolic reactions. It manages both the things, needs of both the reactions. One side for energy, this generation of energy. The other side, yeah, the thing, the for the synthesis of various, biomolecules. That's why the TCA cycle or citric acid cycle, it is also known as amphibolic pathway, because it serves both catabolism and anabolism. It manages, helps in managing both catabolism and anabolism. That's why it is called as amphibolic cycle. So with this, we will stop at this stage and continue in the next class. In the next class, various reactions like glycolysis, then Tca cycle, and alternate glycolytic pathways like Entner-Dudroff pathway, pentose phosphate pathway, then the hexose monophosphate pathway. So all these things, we shall deal, and respiratory chain and generation of ATP molecules. So how it happens, all that we shall discuss in the next class. Especially among all of them, individually these reactions may come for you for the descriptive questions, the last questions where they carry 5 marks things. Some of them, if these reactions are little smaller, they may be asked for the thing, like short notes, or the elaborative for 2 marks or 3 marks they may be asked. That's why, so these, especially more in every year question papers, you come across any one of these different things. For example, they may appear for descriptive things. For example, the TCA cycle, so acts as amphibolic. It is also called as amphibolic cycle. You defined, or this, how, why it is called as so, or otherwise with a neat diagram, discuss the structure and function of electron transport chain. That may be asked like that. Various things may be asked from this metabolic pathway. Metabolism, microbial metabolism. So that we will continue in the next class. So Gavisitesh, Gavisitesh, Gavisitesh.
So we have four cycles in this particular chapter.
Pardon me?
So we have four cycles in this particular chapter.
So I don't know why, it is disturbance. There is. Hello, I can't hear you, Devendra.
Sir, yes, sir. I was asking the in this chapter four cycles are there, na? Yeah, pentose
Yeah,
pathway.
yeah, 4 cycles, Devendra.
Yeah,
So
you sir, you said about yes, sir. Yes, sir.
glycolysis or glycolytic pathway or EMP pathway. That's yeah,
Yes, sir. Yes.
EMP pathway. Next, you have the alternate pathways. Alternate pathways, you have Entner-Dudroff pathway. Then you
Yes, sir.
have the pentose phosphate pathway. It is also called as hexose monophosphate pathway.
Yes, sir.
That is called as. Then you have TCA cycle, tricarboxylic acid cycle. Then
Yes, sir.
lastly, you have electron transport chain.
Yes, sir.
Some text, you can gets the structure, the structure of that.
Yes, sir.
In Google also you will get, with that diagram you should get ready. So from a molecule of glucose, how it results in the formation of 38 ATPs. How at the end of the aerobic respiration, when a molecule of glucose is oxidized to carbon dioxide and
Yes, sir.
it of course, as per the things in information, it results in the generation of ATP molecules. How? You have to explain at a different stage. For example, in glycolytic pathway, what
Eight
is the next
ATPs are
production of ATP? And
eight
say
HP.
all
Yes.
that, then suppose, of course, there is also production of NADH2. And suppose in case of only in case of aerobic organisms, if it is further, if it continues to Tca cycle and ultimately through electron transport chain, that is also realized that NADH2 produce. Otherwise, only ATP in for anaerobic organisms, fermentative or anaerobic respiratory organisms. And in Tca cycle, of course, how many NADH2 and how many FADH2 are produced, all that you have
Three
to,
and two.
six NADH2 and two FADH2 molecules are produced.
Yes,
And
sir. For each glucose, for
yeah,
two glucose units.
yeah. Then ultimately, when all of them have things, at the end of the electron transport things, it
Yes, sir.
yields so many ATPs.
Sir,
And
excuse me, sir. Sir, one more doubt, sir. In that fermentation organism, the NADH is utilized to produce alcohol and energy. Sir, from where they get the NADH? NADH, from only glucose cycle is only operating there, or pentose
actually,
cycle is also.
so in the energy metabolism, there are two kinds, broader kinds. Yes, one is the ATP photophosphorylation. From sunlight, it happens, and in majority, all the heterotrophs, it happens through oxidative phosphorylation. Oxidative phosphorylation, that the common things, which is part of that glycolytic, till that thing, Tca cycle and so on. Whereas in case of fermentative organisms, they think, electron transport, the donor electron acceptor, it doesn't come from outside, it is endogenously managed. For example, in case of yeast, which is an alcohol, which produces ethyl alcohol from glucose molecule. In the first, all the organism, you must remember, all of you should remember, glycolytic pathway is found in all organisms, irrespective of aerobic organisms or anaerobic organisms. But you know, the aerobic organisms, only in aerobic organisms, Tca cycle is present. Tca cycle, Tca cycle is present. Whereas in fermentative organisms, so they, glucose gets converted to pyruvic acid. From pyruvic acid, the diversion occurs. For example, from pyruvic acid, thing, in the middle, acetaldehyde is produced. Acetaldehyde, which acts as electron acceptor to get itself, thing, oxidized to ethyl alcohol, acetaldehyde. Acetyl, ethyl alcohol, and intermediately acetaldehyde. So that means to say, one of the intermediaries of the metabolic pathways takes the role, the as electron acceptor. It accepts to get itself reduced. So acetaldehyde accepts, it gets reduced to ethyl alcohol. So that particular things, so the energy, generated in these organisms, it is only limited, very less. The energy which is released, what is the net production of energy in case of in glycolytic pathway? In glycolytic pathway, it results in two ATPs, two NADH2, and two ATPs. But that two NADH2 that are produced in glycolytic pathway, they are not realized in the form of ATP molecule in case of these organisms, fermentative organisms, because So they only when NADH is passed through electron transport chain, then only it results in the formation of ATP. Otherwise, it is only recycled. NADH2 that is formed, it goes back to NAD thing. It is recycled within. Only the two ATPs that are generated. That is the only benefit in fermentative organisms. But only what is the then why the organisms have to follow this fermentative pathway is it is very important. These for their survival, the organisms need to depend on external source for their electron the acceptors, either oxygen or nitrate or sulfate, which come from the external source. But whereas in this case acetaldehyde is generated as one of the intermediaries of the metabolic pathway, as that is endogenous, it is generated internally. That is the and these fermentative organisms, they survive better compared to aerobic organisms. Aerobic organisms under anaerobic condition, they get eliminated. Next, Devendra.
Yes, sir. Yes, sir. So got, sir.
Okay. So
Yes. Thank you, sir.
thank